about 2 or 3 floors up. And I studied specifically the role of HSD3B7 in the Burmese python liver and how it related to lipid metabolism. So yeah, again, we studied these ginormous snakes. We physically in our lab did not actually do any of the dissections or hands-on handling of the snakes, but we would receive tissue samples from them, from their livers at several different time points to, again, analyze gene expression. The Burmese python is really incredible for a number of reasons. I didn't know much more than maybe a picture when I was younger of them swallowing a huge animal like a little deer or something. But it actually turns out not only do they eat enormous amounts of food in one meal, but then the resulting organ growth that happens, or has actually been demonstrated in the Langwang lab at CU, is just astounding. You can see pictured here the heart and liver, two of the main organs that the Langwang lab focuses on the heart really, and again we focused on the liver, but there's a huge increase in mass very soon after feeding, and it actually decreases very quickly after that a few days later. The biggest thing that the Langwang lab has actually shown, or that helps us, is this discovery that fatty acids in the python's blood, in the blood serum, are actually the circulating factors responsible for this growth. And so if you look at this picture, you can see from the opaqueness of the samples at 1 day post-fed is actually where we see a peak in fatty acid concentration before, again, pretty quickly returns back to normal around 6 or 10 days post-feeding. This is important to note also that if you saw this kind of fat concentration in a human or many other animals, it would most certainly result in lipotoxicity and ultimately death. So the question really becomes, how is the python so able and easily able to efficiently process this amount of fat in food in general? What about its lipid metabolism enables it to do so? In humans and mice, and basically all species we know, the liver is crucial. It's one of obviously the most important organs, especially in digestion. It has a host of functions involving detoxification of alcohol and certain medications or any toxins that enter the body. It also helps to process certain nutrients and store certain nutrients. But what we really focused on, again, is the liver's digestive function in bile production. Bile being the substance that emulsifies and allows for the breakdown of fat in the small intestine and in the digestion system. Here you can see a very complicated diagram. Don't worry about the specifics or any of the words, so to speak. This is just to show bile acid synthesis in the liver is incredibly complex. It involves 17 different genes, or enzymes, I should say, and even more reactions. They can be simplified, though, into the conversion of cholesterol, or fats that have been converted into cholesterol, being converted into bile acids. To excrete fats from the body and cholesterol from the body, you have to first convert these fats into cholesterol, and then they're either converted into bile acids, which are excreted through the common bile duct and stored in the gallbladder before being secreted into the small intestine. You either have to convert them into bile acids, or the bile acids that are converted then bind in the small intestine to the fats and allow for excretion. HSD3B7 is already a known gene, or a gene that encodes for an enzyme in humans and mice that is necessary for bile acid synthesis. It was shown to be necessary in a knockout study done in 2000, where they knocked out HSD3B7 and basically observed a diminishing bile acid pool. From this chart, you can see they graph survival versus time, and you can see the wild type up top, the top curve, is pretty normal, and then there's a steep decline or drop-off in survival rate with the knockout species. This study also actually included, they supplemented some of the mice with cholic acid, one of the primary bile acids, and observed an increase in survival, which is just an interesting thing to note as well. This picture shows a visual representation of a knockout mouse compared to the wild type. The knockout mouse being the mouse on the right, of course, which is much smaller and has actually an interesting phenotype of very greasy or oily fur. So something is clearly very wrong when HSD3B7 is knocked out. Knowing all of this about the Burmese python, about HSD3B7 and the liver and bile acid synthesis, I hypothesized that we would see a gene expression increase very soon after feeding, again, stimulated by that increase in their influx of dietary cholesterol. We tested this hypothesis using quantitative or real-time PCR. We received samples, again, we didn't handle the snakes, but we received liver tissue samples, 2 snakes per time point. We tested about 5 or 6 time points, fasted, 1 day, 3 day, 6 day, and 10 day post-fed. And so we received those samples. We'd have to first isolate RNA using an RNA-seq kit or other methods, and then synthesize cDNA from this RNA using reverse transcriptase. After we were at that point, we actually first, and started the semester, with designing gene-specific primers, because to perform qPCR or PCR in general, you have to be able to amplify a certain target region, obviously our target being HSD3B7. We also did several validation steps, as you can see here, in silico validation and just conventional PCR to make sure that our primers were working initially. And obviously a standard curve production is necessary for qPCR. The results were actually pretty positive. We did see an increase pretty quickly after feeding. It was a little bit later than the initial hypothesis. I had initially thought HSD3B7 gene expression would go up as soon as 0.25 or 0.5 days post-feeding. This was not observed clearly, but thinking back, it actually makes more sense that it would be a later peak. If we think about the fatty acids in the serum, the stimulus for upregulation of HSD3B7 really shouldn't occur until there is that peak of fat and that influx of cholesterol that has reached the small intestine, which should take a couple of days normally. With more time, actually next week, I plan to start investigating other time points, namely 2 days, 6 days, and 15 days post-fed. This would allow, if we look back, kind of fill in some of the between the lines. We see, is there peak actually at 3 days post-fed, or might it be in between somewhere? Also, at 10 days post-fed, there's still quite elevated levels of gene expression. So 15 days post-fed would maybe allow us to see a decline back to normal range. Also, I want to investigate CYP7A1, which is a gene that encodes for an enzyme 1 step previous in the bile acid synthesis pathway. I would want to see also a similar increase in gene expression with that to validate, again, HSD3B7's involvement in the Burmese python. Also FXR, which is the regulator for HSD3B7 gene expression. I would want to, that's a negative feedback loop, so we'd want to see a decrease in FXR expression. A Western blot would further allow us to make sure that functional protein is in fact being translated from our transcript. It's possible that microRNA or the likes are degrading it before it has time for this. So that would again just kind of complete the picture and ensure, or kind of prove more so, that HSD3B7 really is involved in the Burmese python bile acid synthesis. Python lab in general, bigger picture, kind of completing the full complement of genes. The Python lab works to kind of put together this big puzzle for how the python, again, its lipid metabolism process. Everyone in our class is assigned a different gene. I studied HSD3B7 clearly, but many people study completely different genes, not even involved in bile acid synthesis, but involved somewhere else in the lipid metabolism system. And so we want to continue basically to study, and in further semesters and years, study more genes and kind of, again, finalize this picture, hopefully, ideally, at some point in the future. This, again, the whole reason for this study really is with more and more fat and the prevalence of obesity in the US and just the world in general, we're seeing more and more or higher rates of fatty liver disease, specifically non-alcoholic fatty liver disease, which affects 25% of the US population. It's quite nasty, it results in cirrhosis and death. And so this study of the python, we really hope could help us optimize our own biology